So thank you all for joining this global audience today. It's wonderful. And also a special thank you to the folks that filled out the pre-seminar or pre-webinar survey. We've worked today to adjust and augment the content of today's presentation to align as much as possible on some of the feedback everybody shared ahead of time for topics they'd like to hear from us. So thanks again. So on our call today, I am joined by Dr. Ken Lipson. Ken serves as the Chief Scientific Officer and our Vice President of Corporate Strategy here at Ventoux, and he has been a key partner since I started the company. I started the company October of 2022, and Ken and I met in January of 23. So Ken has been a key part of Ventoux Biosciences since the early days of getting going some 20 months ago. Ken brings 30 years of scientific leadership, extensive experience in early drug discovery and target validation. He has worked in a range of therapeutic areas, but has extensive experience in fibrosis and oncology. So, of course, he's been a perfect fit for what we're trying to do here at Ventoux Biosciences and advanced treatments for Dupuytren's and other fibrotic diseases. Ken has over 100 peer-reviewed publications, journals, and book chapters. And you'll be hearing from him very shortly. He'll be sharing updates on VEN-201, as well as our preclinical plan and some of the go-forward plans for us. As I mentioned, I'm Kurt Harrington. I'm the founder and CEO of Ventoux. I spent 25 years in the biopharmaceutical industry. I've worked with large pharmaceutical companies like AstraZeneca and Biogen, and then moved to San Diego in 2005. And since that time, I've worked for small to mid-cap pharmaceutical companies, and also spent a number of years advising and consulting international clients. So for some on the call, Dupuytren's might be new. I think for many on the call, people are quite familiar with Dupuytren's disease, but I wanted to talk briefly about what the disease is, and really this ladders up to what we're trying to do as a company to help patients with Dupuytren's disease. So Dupuytren's is a chronic, progressive, debilitating fibrosis that impacts the hands. It is predominantly fibrotic, but it's also inflammatory and has immune components to it. And currently, as many of us know, there is no cure for it, and there are sadly also no disease-modifying treatments that are approved for it. In a simplified manner, the disease tends to progress most often through stages. As you see on the left-hand portion of this slide, in the early or mild portion or phase of the disease, patients oftentimes appear with a benign tumor or a nodule or a node in the palm of their hands, or pitting, oftentimes below the fourth finger. As the disease progresses, the pitting turns more into cords that start to run up the digits of the hands, which is a collagen-based cord. And you'll see some contracture there in that moderate illustration in the middle. As the disease continues to progress, we see even more collagen formation, and the patients have a severe contracture where the fingers are locked there, unfortunately, and it can go all the way towards the inwards of the palm and be locked there. So that is the disease. It doesn't always progress in this manner, but this is probably more common in terms of the longitudinal flow of the disease. So what's the burden and what's the impact? And I know many patients on the line are very familiar with this, but unfortunately, as I mentioned, fibrosis is irreversible, and it leads to finger contracture towards the palm. It can lead to a significant decline in our ability to use our hands, our hand function, hand mobility. It can impact activities of daily living. It can definitely reduce our quality of life. And I know many folks, unfortunately, that have had to change work or career due to impacts from a work or an economic status. We know that the Dupuytren's can oftentimes lead to pain or tenderness or burning or itching. I will say the literature in this area is underserved, and there's an opportunity there to further characterize the pain, the discomfort, the tenderness that we oftentimes feel as patients. But we do know that patients have, in addition to the loss of function, significant challenges with pain and tenderness. Not everyone and not all the time, but quite often. And lastly, what I didn't capture here is the anxiety and stress that this disease brings to so many patients. I hear from so many patients frequently, which is wonderful. And I think one of the things even I underrepresented early on was the anxiety and the stress this disease can cause for patients, not knowing how fast the progression might take place or occur. So pivoting a little bit, Dupuytren's actually has some really interesting parallels, I think, to rheumatoid arthritis. Both are debilitating hand diseases, unfortunately. Both have a direct correlation with connective tissue in the hand. Both obviously have negative impacts on hand function, mobility, pain, and tenderness. However, where are they different? Dupuytren's has no approved drugs or pharmacologic treatment options that can slow disease progression for patients with early, moderate, and/or even postoperatively for severe patients. Whereas ever since methotrexate was approved in 1988, some 22 different disease-modifying treatment agents are now available for patients with RA. So if you think about it, years before methotrexate, oftentimes patients with RA were treated with surgery or radiation or use of some anti-inflammatories. But now the disease-modifying agents represent about 95% of patients with RA. We at Ventoux are working to develop the first or one of the first approved first-line disease-modifying treatment options to ideally give patients a chance to slow down disease progression and limit the time they might have before needing a surgery, as well as potentially helping improve surgical outcomes postoperatively. So if we say it's pretty common, this probably isn't really needed for the folks on this call, but I do have some folks, as I'm talking about Dupuytren, say, if it's so common, how come I haven't heard of it? I credit Dr. Gary Pest, who I think might be on the line, Dr. Charlie Eaton, and Dr. Keith Denkler, some of our renowned hand surgeons in the U.S. treating Dupuytren's, for an article 2 years ago where they referred to Dupuytren's as the most common crippling hand disease that we haven't heard of. I love that nomenclature, and it's exactly right. I oftentimes say that it hides in plain sight, but it is extremely common and prevalent and progressive. So these folks here are just some folks that we know of that are more in the public eye that are impacted by the disease. I think John Elway, in his commercials previously has spoken a lot about he actually had to leave the NFL and his role with the Denver Broncos due to an inability to hold the ball any longer due to his Dupuytren's. And very recently, actually, I think a week ago, lower left, John Bonzal from the Oak Ridge Boys was also a Dupuytren's patient and unfortunately recently passed away. So I wanted to recognize him since we lost him recently, and I grew up listening to the Oak Ridge Boys as a kid back in the day in Georgia. So it definitely impacts a lot of us, whether we're celebrities or not, and we must find better options to treat this debilitating condition. So why are we working on this? Why are Ken and I and our advisors working on this? I'm one of the millions of patients affected. In the U.S., there's about 6 million patients that are projected to have Dupuytren's disease. So the prevalence is similar to that of Alzheimer's disease in the United States. My dad is here on the upper right-hand photo, and I'm left, and then my 2 boys are on the bottom. So I started this company with the personal mission to change the course of treatment options and ideally bring in novel treatments that we could have for early to moderate disease to slow this disease down. My dad and I together have had 11 different surgeries. I had 9 different needle episiotomies, 2 open surgeries. My dad just had a fasciectomy, which is the larger, more invasive surgery where they open up the tissue and take out the fibrotic tissue. And then I've also personally had 3 rounds of targeted radiation. So I am grateful and thankful for my radiation oncologists and my surgeons. And I think all the surgeons I know of that I've spoken with that treat Dupuytren's a lot are still in support of new and novel treatments that can help slow this thing down before the patients need to get to them for different surgical interventions. So it's a personal mission for me, for my family, for the fellow millions of people around the world that have it. And also, should future bloodlines of the Harrington family get it, I wanted to make sure I did everything I could to try and slow it down. So I'm grateful for the team that we have in place that's helping us do the work and move us forward. Really quickly, we talked a little bit about treatment options. Unfortunately, for those of us that know the disease well, there just is not a lot of options. Very oftentimes, and the literature supports it, and I lived it, you go to hand surgeons early on and you're instructed to watch and wait and come back when you have a 30 to 40 to 50 degree bend and have a surgery. Radiation therapy is emerging. I still say oftentimes it's patient-led. That said, there are surgeons very much so advocating it or using it as part of their treatment for patients. But surgery to this day really remains the mainstay of treatment. Fasciectomies, needle episiotomies. Unfortunately, you'll see amputation here. Amputation is one of the most common elective reasons for Dupuytren's patients outside of like somebody in a car accident or an ER. So unfortunately, patients with Dupuytren's will self-elect to have digits removed, either out of frustration due to the limitations they've had or after multiple surgeries, and they need to make a different change for their life and their quality of life. So we're looking to augment this and provide some new and novel treatments to the table. So again, we're looking to fulfill this disease-modifying space where we can offer novel solutions to slow the disease down. So Ken's going to talk more in a minute about the mechanism of action and our specific data thus far. but our macro vision that we're looking to enable and the goalpost that we really put together as we targeted screening different drugs heading into this a year and a half ago is to really be able to identify a molecule that's safe, as safe as we know, and potentially effective to slow or reverse the progression of the disease. This is, as I've already mentioned, it's a pretty stark departure from the current options, which again lead towards later stage patients and watching and waiting. So our approach is for a lead candidate that would be a first-line disease-modifying, disease-slowing treatment that could work across all stages of the disease. And I'll talk a little bit more after Ken's session on what we envision as a target product profile as we work through the different development stages. So I've mentioned Ken, introduced Ken. You'll hear from him in a second. Who else has been helping us in this endeavor thus far? We have put together a wonderful world-class ensemble of advisors that have helped us to get to this point so far. First and foremost, Dr. Keith Denkler out of UCSF with a private practice up in Larkspur. Keith is an internationally recognized key opinion leader in surgery. He's a plastic surgeon with a hand fellowship, and he alone has treated over 10,000 fingers with needle aponeurotomy, and was one of the leading surgeons helping bring that surgical procedure to the U.S. from France. So Keith has been a wonderful supporter and advocate and advisor since inception. Dr. Lata Satish is early stage and has spent her career working on understanding skin inflammation, infection, and fibrosis with a keen focus on molecular pathways and what drives Dupuytren's disease. She's also done extensive work herself looking for small molecules that could potentially help change the course of this disease. And Lata is also recognized as winning the International Dupuytren's Best Paper Award in 2016, so brings a lot of early stage development experience to us on the team here. And then lastly, but not least, is Daiva Bayarunas. Daiva is an endocrinologist by training. She's been in the industry for over 25 years. She's previously served as chief medical officer. So she brings extensive leadership experience in R&D and clinical development, really from phase 2 and later, but obviously she can help out across the entire continuum. And she's also worked across a number of different formulations and routes of delivery. And lastly, she is a very strong advocate for Dupuytren's and emerging and novel treatments to help change the course of this disease. And my last slide before shifting over to Ken is really what has been our strategic approach as we've tried to find new treatment modalities. There is not an established animal model for early disease to evaluate drugs in. Dr. Satish has actually developed the only known animal model to date. Her model actually induced contracture, so it was a little bit later stage model in mice. So given there's no animal model to leverage for a early development of a novel modality, we looked across a number of different angles, but we've decided and landed on the best strategic approach to identify options for Dupuytren's as quickly as possible to get treatments to market as quick as possible is via drug repurposing. And simply stated, repurposing is leveraging another existing compound to treat a new condition. This route is faster oftentimes because you can leverage the existing reference products data in your application. So there are data that is already meeting the needs of the FDA that you don't have to do a unique study for. So you can be quicker. By being quicker and having some reduced studies, you can be more cost effective, which is helpful because we bootstrapped this company for the first year until we had some wonderful friends and family advisors coming on. And it can be less risky because you know a lot more about the compound. More times than not, it's been through phase 3 trials and in market. A couple quick examples of repurposed medications just for reference. Rituxan used to be the #1 selling oncology product for non-Hodgkin's lymphoma. Interestingly enough, as patients were getting their Rituxan for their NHL, those that also had rheumatoid arthritis noticed improvements in their RA. So a repurposed indication for Rituxan was going from non-Hodgkin's lymphoma into rheumatoid arthritis. In addition, Botox, I think one of the most successful products in history, interestingly enough, started out as a rare eye muscle disorder product. And then, of course, now we know Botox is used for dermatology, overactive bladder, chronic migraine, and I think 21 different indications. It's had an incredible clinical program. And then lastly, also one of my favorites is Viagra actually started out as an angina product. So they were initially, Pfizer was looking at products for angina in the cardiovascular space. And then as a serendipitous side effect from the initial studies, they noticed it actually worked on ED. So that was a large pivot, but of course, a repurposing example as well in that of Viagra. So just a few examples, but that really encapsulates our repurposing. So we're going to talk more about the molecule here in a minute, but our platform is to repurpose VEN-201 to leverage it for Dupuytren's patients and ideally move to market as quickly and safely as possible for patients. And with that, I will pass it over to Dr. Lipson to carry us forward.
Hi, everyone. I think you can go to the next slide. So we chose VEN-201 as a clinical candidate based on a variety of different data, some of which was in the literature, including pathway analysis that suggested that its target was likely to be useful as an to inhibit in fibrosis, as well as some target validation studies using both genetic and pharmacological approaches that showed inhibiting it either in kidney model or lung models was efficacious, as well as some mechanistic studies that showed that it could change the biology of myeloid cells like macrophages and mesenchymal cells like fibroblasts and myofibroblasts in a way that would be useful for fibrosis. But relying on the literature alone, it can be risky. And so we decided to run an experiment and test it ourselves to confirm that we could see antifibrotic activity. And if we go to the next slide, go ahead, Gert, next.
There we go. Sorry, a little delay.
I will first introduce you to some of the participants, and then later I'll show you the data from that study. Now, we're going to start at the lower right corner. You see the myofibroblasts down there. These are cells who has a primary function in wound healing. So they're contractile, they help bring wound edges together, and their main function is to lay down extracellular matrix and remodel it and rebuild the gap in the tissue that's caused by the wound. They can be generated from a variety of different cells. On the slide, I'm showing fibroblasts or other local mesenchymal support cells, as well as epithelial cells. They can also be generated from bone marrow-derived cells that are thought to be related to monocytes that get recruited to the wounded tissue and then can transdifferentiate into myofibroblasts. Now, you can think of the myofibroblasts sort of like the road crew that comes to fix a pothole, for those of you who aren't scientists. When they're needed, they show up, or they hopefully show up, and fill in the hole and make it smooth again. And supposedly then the road is fixed. But if you think about what would happen if the road crew didn't stop working when the road was smooth and fixed, and they kept putting down more material and building it up, all of a sudden you'd have a bump in the road instead of a nice smooth road. And then think further, what would happen if 5 different crews showed up, and since they had to get work to get paid, they started laying down extracellular matrix or road material, even though there wasn't a problem. And before you know it, the road looks like a mogul field on a ski slope full of bumps, and the road doesn't work so well anymore. That's essentially what happens in fibrosis. The myofibroblasts get amplified. There are too many of them. They don't go away. They keep laying down extracellular matrix, and it interferes with the function of the tissue. And that process, in essence, is fibrosis. Now we're going to move to the left side, and where I'm showing some myeloid cells. So monocytes on the far left, MO, differentiate into macrophages. And macrophages come in different activation states. And those different activation states have different functions. So M1 macrophages, for example, have a primary responsibility to watch out for infections. So when a pathogen like a bacterium gets a foothold in the tissue, the macrophage will secrete chemokines and cytokines to recruit other immune cells to the area to help fight the infection. They also secrete reactive oxygen species that can directly kill the bacteria. but these reactive oxygen species are not specific. And so besides killing the bacteria, they can cause tissue damage. So between the inflammation and the tissue damage associated with the reactive oxygen species, the signals can promote the formation of myofibroblasts because there's a problem that needs to be fixed. Below M1, you see M2A macrophages. Their primary function is to initiate tissue repair. To continue the analogy of the road repair crew, you could think of this as the foreman who comes and tells the workers to get started on repairing the road. Now, the M2A macrophages promote the formation of myofibroblasts primarily through secretion of TGF-beta, which is a very pro-fibrotic cytokine, but I'm sure they do other things as well. Now, below the M2A, you see the red stop sign M2C macrophage, and its primary function is to resolve tissue repair. And it does that by secreting cytokines like IL-10, which help to deactivate the myofibroblasts and allow them to stop laying down the extracellular matrix. You could think about them as sort of the inspector who comes and takes a look at the work that the road repair crew did and said, okay, guys, looks good, time to go home. You know, hopefully they go away, but if they don't, then you end up with the problems that I spoke before. Now, I'm telling you all this because the primary target for VEN-201 is actually found on all of these cells. So when we add VEN-201, several things happen. First of all, we prevent the formation of myofibroblasts directly by interfering with, for example, conversion of fibroblasts to myofibroblasts. And we promote the resolution of the myofibroblasts. We also interfere with the formation of both the pro-inflammatory M1 and the pro-fibrotic M2A fibroblasts. And when we reduce the numbers of those, we reduce the number of subsignals that are stimulating the formation of myofibroblasts. In addition, VEN-201 will help convert the M1 and the M2A to M2C. So when we reduce the M1 and M2A and increase the M2C, that further helps shut down the myofibroblasts and push the system back to a normal homeostasis, back to a normal condition. So we're having both direct and indirect effects on the tissue repair to restore what should be more normal and hopefully shut down the fibrosis. So next slide. So we don't expect to actually cure Dupuytren's disease. Unfortunately, there is a genetic predisposition. Not everyone gets it. It tends to be most commonly occur in people of Northern European background. So there is some kind of genetic predisposition. It hasn't really been established exactly what that is. But on top of that, there are probably also triggers that promote progression. For example, just using your hands may be able to promote it in some way. So we probably can't cure it, but what we should be able to do by knocking down the active myofibroblast is slow extracellular matrix deposition pretty significantly. And by doing that, prevent the contracture that causes so many hand problems. As Kurt will explain in a couple of minutes, what we're targeting to do is to be able to treat only a few times a year so that there isn't a tremendous burden on patients here. And that would be the goal with using a long-acting drug release approach. So if we go to the next slide, I will show you a little bit of the data. First, I'll explain the experiment. So to make sure that VEN-201 actually had antifibrotic activity, we used a bleomycin-induced dermal fibrosis model. Now, bleomycin is a chemotherapeutic agent. It's pretty toxic, which is why it's used to kill cancer cells. One of the side effects of bleomycin, though, is to cause fibrosis. And once that was understood, it became a useful laboratory tool to cause tissue damage and to cause fibrosis. In this case, we're injecting bleomycin daily, just below the skin, subcutaneously, into mice for 28 days. And then we treated those mice with either nothing for control or with VEN-201 or VEN-202. Both of those agents were supposed to be antifibrotic based on the literature, but we didn't see actually any activity with VEN-202, and so we've dropped that from our portfolio, and we're moving forward with VEN-201 because of the data that I'm going to show you on the next two slides. So what we saw is when you treat with bleomycin, if you look at the histology on the right side of the slide, the top two sections are stained with H&E, hematoxylin, and eosin, and the bottom two with Massen's trichrome. So on the left column, it's bleomycin with vehicle, on the right, bleomycin with VEN-201. So you can see upper left quadrant that the epidermis marked with an E is pretty thick, and the dermis looks kind of chaotic, that's marked with a D. And if you look on the right, the epidermis looks much thinner than it does with bleomycin alone when we treat with VEN-201. And the dermis looks somewhat more orderly. And now if we go to the Massen's trichrome stained tissues, MT, nope, let's go back, sorry. The Massen's trichrome stains collagen blue. And so you can see on the left, the blue is much more intense. You can see some light blue in the epidermis, and you can see some very thick cords, for example, that shown by the arrow. Whereas on the right, when we treat with VEN-201, there's much less blue in the epidermis. There's much less intense staining in the dermis, and the collagen fibrils that you can see tend to be thinner. Okay, now we can go to the next slide. So to quantify the amount of fibrosis, we use digital imaging processing. And basically what was done was every pixel in the slide was examined. And if it was blue, it was turned into the green mask that you see below. So on the left column, it's bleomycin treated animals. On the right, bleomycin animals then treated with VEN-201. And obviously, it's very clear you can see a lot less blue with VEN-201. And on the right, in the bar graphs, we actually show big reductions in collagen deposition in both the epidermis and the dermis. And so, again, we were able to confirm that VEN-201 actually had antifibrotic activity, even in the face of a very aggressive model. And so we were confident to move it forward as our clinical candidate. So Kurt will now explain our
Thank you, Ken. So thinking forward and moving back, I always speak in terms of like, what does a target product profile look like for us? And that's really when we get through all the key stages of product development, what might that product actually look like when it's out in the real world for physicians to utilize for patients like us? So on the right-hand side, the second, third, and fourth bullets really bring that to bear. As we've talked about, we're looking to create a long-term, novel, disease-modifying treatment that can either manage or delay the progression of the disease. We're looking, as Ken mentioned, in a long-acting injection. I would love to have an oral. I think most patients would prefer an oral. This disease is complex. The experts believe it's 20 to 50 genes that are involved. Targeting locally likely gives us the best chance of success, at least in our belief at this time. So we are working on a long-acting local injection that would be administered by a physician, either a hand surgeon or a rheumatologist who also treats disease-modifying other conditions like RA. We're targeting either every 2 to 3 months administered by a physician. But what does that mean? We don't at this point believe that this would be ideally, it's not a chronic for the rest of your life treatment. Our plan and our hope would be that we can inject into our nodules for a year and a half, a couple of years, and ideally we see enough softening of the early nodules, erosion or disappearance of those early nodules, that then we could ideally stop therapy for a while. Should we have another nodule pop up somewhere else, we could treat it, so on and so forth. But we're on a macro level planning for a physician-administered injectable that's administered every 2 to 3 months, depending on physician-patient discretion, is also contingent upon our future studies and what the data tells us. We are targeting 2 different indications. We've talked a lot about the mild to moderate patient. So we see this as a potential drug that could be used as monotherapy early on, really at the first sign of a node or a nodule or the first sign of a cord. In addition, given the recurrence rates that end up high after procedures, we are also very interested in a separate program that could be used as an adjunct to surgery. So you think about a patient maybe has a fasciectomy, and then a week to 2 weeks later, they're administered VEN-201 to ideally help really preserve the magic of the procedure administered by the surgeon and further reduce recurrence and ideally lock in those outcomes for patients. And up at the top now, to go in reverse order, just to speak a little bit, but we haven't unblinded the molecule yet. We're still just referring it at present as VEN-201, but it is a small molecule. It is not a peptide or a biologic. It has been approved already and is now generic, but it's been approved previously in over 90 different countries. It's been around for over 20 years, I should say, with an established safety profile, and it has no black box, which is one of the more severe warnings and precautions we oftentimes see in package inserts. So obviously we need to do our own work and test it in hands and in Dupuytren's going forward, but it's a safe and effective molecule in its current form, which gives us increased confidence to take this one forward around what it could do and what it may do for patients. Okay, so pivoting quickly to the what's next. Near-term priorities for us are really fivefold, and we tried to macro list them here. First and foremost, it is engagement with the FDA through pre-IND meetings. While we are repurposing a drug that has been approved and used for 20 some odd years, the pathway and the clinical development program we're treating is really the first of its kind. So working with the FDA to share what our plans are and hearing their input to ensure that we put together the optimal clinical program to meet the targets we're working towards. So a key element is the pre-IND meetings and initial engagements with the FDA. We've started some of that, as you can see from the initial checkbox, but there's more work to do. In addition, in the study that Ken went through, that was a once-a-day injection that we formulated for that study. As I just mentioned, we're talking about longer-acting injectables. So a key element at this phase is the formulation planning and development and screening work. In addition, the intellectual property and portfolio strategy is key. We've already filed for a 20-year method of use patent application in fibrosis, but of course, to follow that would be composition of matter as we start to further advance on the formulation development. To do all of this work, a lot of our current focus is raising funds at present, either through venture capital firms, angel investor, private investors, or we also have a crowdfunding equity-based program. If anybody wants more information on that, there's an investor button on the website. Happy to talk offline, but those are key priorities currently. And then, of course, building the additional rest of the team out to support this next phase of work, because Ken's really good and our current advisors are really good, but we need more manpower and support to get it done. So that'll be part of the process. And as part of that, we are very pleased to announce today the onboarding of 3 new wonderful advisors to help us with this next stage of the company. Dr. Reddy at the top is a career formulation CMC expert, knows Ken from way back, and I actually met Ken through Latchma, so it's wonderful to have him on officially as an advisor, but he's spent an extensive amount of time developing formulations and screening, so as we're focused on this next stage of a long-acting, Latchma's going to play a key role. Matt Kravitz, I've worked with for a long time, one of my favorite biostatisticians I've ever worked with. We worked together back at Biogen working on Rituxan for RA. Matt currently is the SVP of biometrics at Gossamer Bio, and he'll play a wonderful role as an advisor with his experience as a statistical lead as we look to plot out our study designs, the applications, and start further engagements with the U.S. regulatory bodies as well as international regulatory agencies. And then Johnny Randall is a 30-year plus executive in finance. Johnny's been a CFO, excuse me, for both private and public companies, as well as help companies go from private to an IPO. So Johnny brings a wealth of institutional financial expertise to the table, and will be very helpful as we go forward with investors and advisors, and so on and so forth. So very pleased to announce these three, and so happy to have them helping us here now at Ventoux with the rest of the group. Okay, one last slide before we pivot to some questions. And this one, I'm taking my Ventoux Biosciences hat off just for a second. This slide is more about the need to align more if we can, and further activating ourselves to raise awareness and educate and advocate for this condition. So these are some bullet points, just some ideas that I thought I'd share, that if we can further align our voices and raise our voices a little bit, I think it's very helpful as a Dupuytren's patient myself, and I know for the folks that I meet with regularly. So just a couple ideas. One, I know there's a lot of wonderful patient groups out there. I'm in a lot of them, and I greatly value them. Since forming this company, I've had the wonderful pleasure of meeting with patients almost weekly that reach out, call, and I can't tell you how much that's been wonderful, just to sit with other patients, share stories, testimonies of what we're going through, our treatment, our experiences. So I would encourage any on the line that are patients or loved one of patients to encourage not only the online, but to go have a coffee or a tea and just chat if you can. In addition, I put Speak Up here. The literature is not great. I think many of us know that in terms of how we live with this disease and how it's characterized in a lot of research. We know oftentimes there is pain. We see it listed as painless. To me, there's a much better way to characterize what we're experiencing if it relates to pain or tenderness or itching. So here I would say, as comfortable, be comfortable sharing what you're feeling as much as possible with your appropriate physicians. In addition, there's some wonderful advocacy and other research groups out there, the International Dupuytren Society, the Dupuytren's Research Foundation here in Florida in the US, and the British Dupuytren Society. I would say if you're not involved with them, reach out, see if you can help get involved. They're wonderful and great people. A couple more that are a little bit more in our tent, but still not directly part of Ventoux or unbranded. We have a new Hands of Change podcast. This is a podcast that Ventoux sponsors and supports, and I'm the moderator, good or bad. But this is the whole idea here is really patients sharing their stories, their experiences. We've got treatment providers coming on, from radiation oncologists to surgeons, as well as researchers in the space. So it's a way to listen and learn from others. And then we also have a newsletter on our website. We try and put out not just company news, but relevant Dupuytren's information. So if you're interested, please opt in and join our newsletter. And then lastly, given the need to raise awareness and shift this from being the most common crippling disease we haven't heard of to the most common crippling disease people have heard of, we've started another little option for patients to get involved. This is directly, there's a link currently on the website if you go to VentouxBio.com, and then in news and events, we've just launched a pop-up store with Dupuytren's information. So a lot of the content is, erase Dupuytren's disease, if you will. So it's more specific to the disease. There's also some items around Hands of Change and the podcast, and ideally kind of that idea or that movement. And there is some information or some content that also includes branding and things like that from Ventoux Biosciences. So just some ideas to really try and educate, support each other, and raise awareness as we move forward with this, and as we want to look to try and change the game with some novel treatment modalities for the market. So with that, I will say thank you on behalf of myself and Ken. And just to recap, we obviously feel this is a significant unmet need. We believe VEN-201 is a novel first-line, potential first-in-class treatment. We've built an experienced and accomplished team, and that's growing. And we're pursuing an efficient and expedited development plan to ideally help patients as soon as we can, but again, in a safe and effective manner. So with that, we'll pop over to address some of the questions. So if you have a question and haven't posted it yet, please go ahead and do so. And I will go to it. Looks like John Foley says, please don't do background music. What if John knows what music I like, so that's probably why he suggested that. Noted, John. Got it. Thank you. And then Justin says, cycling likely caused it for me, 44 years old, aggressive DC. Yeah, so sorry to hear that, Justin. I think we've talked offline as well, and I used to mountain bike as well, and I think that probably didn't help. I also played sports and broke fingers and things like that. And there's definitely literature around the vibrations and some of those things that can cause the trauma and make it worse. One thing I would suggest now is a triathlon bike, because you ride on your elbows versus directly on your hands. Let's see. Any other questions from anyone? I don't see any. So I will wrap it up here. And if you have questions after our emails are posted, our information is posted, we try and be as responsive as we can. So please reach out. Thank you, everyone, for attending. We welcome feedback or questions for our other talks as we go.
Actually, Kurt,
Yep.
Julie just had a question, I believe.
Hi, Julie. How are you? If a person has had more than one NA, will this be helpful over the long haul? We hope for it to be, given depending on the stage of the disease, but that is very much in line with our vision of this as an adjunct to either a fasciectomy or even post as an NA. To some extent, thinking a little bit of it, how radiation can be used now, whether it can be used early by itself or later on as an adjunct. We'll obviously need to see how the clinical data bears out, but that is part of the plan. Let me mark those. How quickly do we think this will be available from Hillary? Our current plan, Hillary, and this is based upon the current fundraising we're doing, the first study in humans, which we're proposing to be a phase 1b study, we're currently projecting that to be about 24 months out once we do the formulation work and the FDA work. And then from there, we'd have to see what the phase 2 and or phase 3 trials look like. Sometimes with a 505b2 filing like we're doing, depending on how much dose ranging and things like that are in the 1b, you can move straight to a phase 3, but we'll need to wait and see on that. So I would say the first projected timeline to patients in a phase 1b would be approximately 24 months. Let me see. Answered. Answered. How do you see trials being done? Neither radiation oncologists or hand surgeons do not do. Bob, the last part of your question fell off. I'm going to jump back. So Bob, if you can repost that, that'd be great. The last part of your question fell off for me. Anonymous says, the Dupuytrens usually begin in the hands. Yes, so Dupuytren's has a lot of sister cousin conditions, but Dupuytren's is the fibrosis that occurs in the hands. It typically occurs first in the palm of the hands, but not always. On my hands, my right hand middle finger is my worst digit, and I never had a nodule that started in the palm. My middle finger was actually, I started to get a cord in the MIP joint. So it doesn't always follow the nodule to cord. Mine started with a contracture. In the foot, there's a disease with almost the exact same pathophysiology called Lederhose. And then adhesive capsulitis is in the shoulder or frozen shoulder, and oftentimes, referred to as Dupuytren's of the shoulder or Dupuytren's of the foot. And then unfortunately, men can get the same thing in the penis, and it's called Peyronie's disease or penile fibromatosis. So there's several different fibromatosis that can occur, but in the hands, it is called or known as Dupuytren's. And let me see. Anonymous, we answered that one. Will this help slow progress if you had amputations? Great question for Rudy. Ken, do you have any thoughts on that at present?
Well, I think it will depend. If the finger has been completely amputated, I don't know enough about how the disease would then manifest, but it would help potentially with other fingers.
Yes, yeah, that's a great point. So if you had a digit amputated, unfortunately, but you have the nodules appear or Dupuytren's appear in another palm or another digit, we would plan on this being utilized for those areas to ideally help out. I hope that answers the question, Rudy. Great question. Hey, go ahead.
And I think Bob finished his question.
Oh, he did. Let me scroll back
a few lines up.
Okay, looking for Bob.
Basically, he's saying that the surgeons, the radiation oncologists don't do administration of treatments in general.
Yes, no, for sure. Yes, the hand surgeons do. So you think about the current surgeons now that would administer Xiaflex, which is approved and more for a severe patient, almost like an alternative to a needle episiotomy. So we know the hand surgeons, whether it's a plastic or an orthopedic, are doing buy and bill administered treatments for the hands. So we would follow something very similar with Xiaflex, or those that are using a non-steroidal anti-inflammatory as well for Dupuytren's. And then for rheumatologists, they of course treat a wide array of patients with biologics, with injectables, with infusions, and they buy and bill. So we would see this as a specialty product administered predominantly by surgeons and their care team, NPs, PAs, or rheumatologists and their care team, NPs, PAs. And then potentially, as we grow and evolve and work on education, this could be something potentially administered with upper decile internal med or potentially dermatologists. But I think at present, our core focus is really on continuing with the surgeons, since they end up seeing so many of these patients to begin with, and then really working to partner and grow with rheumatology.
So Justin was asking about full thickness skin transplants and if it could be used in conjunction with them. I don't know enough about this topic to be able to answer well, but I will try to point out that the fibrosis, it really begins in the palmar fascia, which is very deep. It's below the skin. It's like a thin layer above the tendon and below the skin. So I don't know exactly how much of the skin is replaced and whether this would have any effect, whether it would have any effect on the progression of the disease or not. I would have to read more to understand more.
Got it. Thank you, Ken. And then Dr. Satish is on and listening, and she sent a note through to follow up on Rudy's question. And Dr. Satish also says it also depends on how aggressive the disease is, as a follow-up to Rudy's question. Trials are focused on hand surgery. Yes. Let's see it. Go ahead,
Hillary,
Ken.
about signing up for a clinical trial.
Oh, so at present, we aren't enrolling in the clinical trials just yet. This next phase is really focused on the formulation and the FDA work. But that being said, I would encourage everyone interested in follow-ups in general, as well as we advance towards clinical trials, to just opting in on ventousbio.com for our updates and our newsletters. As we do advance in time, we'll plan on doing some outreach around what the study might be, what the sites might be, and some of those things through all the proper channels. So if you're interested right now in just updates and things, that's the best place to go. And I am looking for any more. These are great. So thank you, everybody, for sending them in. Thank Justin for the additional links. Those are great. And Hillary said, oh, okay, 2. Contribute and sign up. Yeah, so you can opt into the website to opt in for communications. And then if folks are interested in contributing to the crowd equity. So if you go to investors, the investor tab on the website, that'll take you to 2 options. One is a follow-up meeting to talk with more institutional investors. But we do have an equity crowdfunding campaign, which people can put in as small as $100 if they want to contribute towards the work we're doing as well. So we kind of have a 2-pronged fundraising strategy at present to try and engage as many folks as possible. And of course, anything on the store is going to Dupuytren's research as well that we're leading. So let's see. I think, did I get to all of them, Ken? I'm trying to improve my skills here at
I think so.
skimming all the questions. And thank you, Justin. I appreciate it. Thank you, everyone. We appreciate follow-up on this. If you want another call like this or different topics as we go forward, we are all in, and this means a lot to us. So appreciate everybody listening. Appreciate everybody joining in, and thanks a bunch. So please stay in touch. Please follow along if you can on the website and through our newsletters. And thanks again, Ken, for joining. Wonderful job.
My pleasure. Bye, everyone.
Thanks, everybody. Bye-bye.